Hi everyone, and welcome to the webinar on retrieving biological data from EMBL EBI resources using DBfetch. My name is Nandana Madhusudanan, and I work with the web production team at the EMBL EBI. Along with me, my colleagues Fabio and Adrian also joined the call, who will be available in the Q&A box for answering your queries during this webinar. In this webinar, we will see a brief introduction about the DBfetch, how to retrieve entries from it, the databases available via the DBfetch, and various output formats of the service. We will also cover how you can access this service programmatically, which we will be demonstrating using the Python client. As you all know, the European Bioinformatics Institute provides access to a wide range of biological databases and analysis tools. You can access our services either via the web interface or programmatically using the RESTful web services. So this is the service page of EBI. Here you can search for your resource of interest and go to the respective resource or tool page. We have more than 40 data resources and several data analysis tools listed here. If you're looking for a single point of access to retrieve entries, you can use our DBfetch service, which stands for database fetch. You can access data from majority of databases hosted at the EBI using this service. This is the homepage for DBfetch. You can access it from the EBI service page or can access directly from the URL www.ebi.ac.uk/tools with a capital T/DBfetch. There are mainly 4 fields or parameters that you need to specify while using the DBfetch service. They are the database, entry or item field, where you specify the accession number or entry identifier, and the format and style. The database and the entry fields are mandatory, whereas the format and style are optional parameters. If you haven't specified this, it will take the default values. You can get a single entry or bulk of entries from DBfetch. As I mentioned, you need to provide the database name and the entry ID or accession. As you can see here, either provide the identifiers or accessions in the search item box or use the file upload option. If you're specifying the entries in the search box, first choose the database and then provide the entries in the search item field. If you're providing multiple entries, separate each search item with a comma or a space. If you're using the file upload option, provide the entries in database name :ID format, and each entry should be on a new line, and all belongs to the same database. Note that you can get maximum 2000 entries in one go. A few of the databases from DBfetch are mentioned here. We have structural, patent, protein, and nucleotide sequence data. Once you provide the database and entry, choose the output format in which you need the data. Different databases store data in different formats. If you haven't selected any, select the default format, which is often the default format for the specified database. An example, like if the default for the ENA database is EMBL format. So the selection of output format varies based on the database. As you can see here, for the database ENA, these are the output formats, and for the database PDB, the output formats are a bit different. These are a few file formats supported by DBfetch. FastA is one of the most common and simple sequence format. It starts with a greater than symbol with a description line, which is the header, followed by the sequence. The EMBL format has both sequence and annotation. As you can see here, the EMBL format starts with an identifier line, ID, followed by several annotation lines. The sequence starts with the SQ line and the file ends with a double slash. The Swissprot is similar like EMBL format, but is for protein sequences. And the PDB file format describes the three-dimensional structure of biomolecules. You can see example PDB and Swissprot format from the links mentioned in the previous slide. Another parameter in DBfetch is the style that refers to how entries are rendered in the browser. The results can either be delivered as a raw text or as HTML. HTML has markup, while raw is a plain text. Specify here which style you prefer. The default is almost always HTML. For more information about the databases and formats, you can check this page. This includes a short description of the database, some example identifiers, and details of the available data formats and result styles. Now we will see a quick demo. So this is the home page of DBfetch. As you can see here, you can select your preferred database. Suppose if I want to retrieve an entry from UniProt KB, specify the entries here. If I want multiple entries, separate it with a comma or a space. Specify the output format that you want and the style. Now you get the UniProt IDs WAP-RAT and WAP-MOUSE in FastA format. If you haven't specified any format, it will select the default format for UniProt KB, which is the Swissprot format, and give you in HTML style. From here, you can go to the respective database entry. If you are using the file upload option, mention the entries in database name :id format, as I mentioned before, and each entry should be in a new line. And all the database on all the entries should belongs to the same database. In this case, we have selected the UniProt KB database. Suppose if I am mentioning the format as EMBL, when you try to retrieve, it throws you an error. It says that EMBL is not a supported format for UniProt KB. So you need to choose the format which suits the database that you opted. For more about the database styles and formats, you can go to this database documentation page, which has the aliases for each databases. And each database section, in this case, this is for the ENA sequence, so has its corresponding format, style, and sample identifiers. Here, you can also see the data resource from where you got this data in DBfetch. For some databases, it also mentioned the version here, so which helps you to find the different versions of that entry. Now, going back to the presentation. If you want to use the DBfetch for direct access, you can make a scripted HTTP request to DBfetch, which is very simple. You just need to specify the database, ID, format, and style if needed as query parameters. That is, each parameter is separated by an ampersand. Like in this example, here you can see the parameters, database and ID, which are separated by ampersand. If you want to mention the style here, the format here, you can specify the format. Likewise, you can specify the style as well. And you can provide multiple entries if you want. So for more about the DBfetch URL syntax can be found in this help page. If you have too many entries, it would be difficult to mention the IDs in the URL. In that case, you can go for the programmatic approach. Programmatic access is a way in which the data can be accessed from a service using some clients written in several programming languages. We have Perl and Python clients for DBfetch, which is freely available in our GitHub. Links to both Perl and Python clients are provided here. Now we will go through how to use a Python client. Note that for using the Python client, you need to have the Python version 3 or later, also XML Trump 2 and requires Python modules. If you don't have the modules, you can install it with pip install command. So for using the Python client, you first need to download the client, and which is available in our Git repository. So I am going to download this DBfetch Python client here using the wget command. Now I have the dbfetch.py Python client. If you want to see how to use the client, run it using -- help option. So this will give you how to use this client. So there are a number of methods available, which helps you to find the available databases, formats, and styles. Now I will show you how to get the available databases from DBfetch. So if you provide the get supported DBs method here, it will list all the available databases in DBfetch. Likewise, if you want to get the formats, specify the get supported formats. This will give you the database and its corresponding formats, output formats. Suppose if you want to get a format of a particular database, you can use this method, which is get DB formats, and then specify the database. So this will give you the output format for Uniprot KB. Now we will see how to retrieve data from DBfetch using this client. So we have two methods available for that, fetch data and fetch batch. Fetch data is used when you need to retrieve a single entry, and fetch batch is for multiple entries. Suppose if I want to retrieve a single entry from Uniprot KB, specify it like the database :ID, then the output format that you needed. Similarly, if you want to retrieve multiple entries, you can specify the database, then the IDs, and the output format. In case you want to use the file upload option, you can do that using the using the fetch data method. Okay. So I have a file which has 2 entries from Uniprot. And if you want to retrieve these entries, you can use the fetch data and then use ampersand. So this is how you retrieve the entries. So I'll highlight the commands. So this is for retrieving multiple entries using file upload option. So the method will be fetch data and specify the file name. Before that, you need to mention an @ symbol. Then for batch retrieval, you can use the fetch batch method and specify the database, then the entries, and then the format. For a single entry, you can use fetch data method and the database : the ID, then the format. So these are the commands that we tried in this demo. DBfetch is developed and maintained by the web production team. If you have any questions, please contact us at support@ebi.ac.uk or via feedback form. You can also check our documentation and FAQ pages for any help. Thank you.
So with that, I'll hand over to Fabio and Nadna for Q&A. So I think some of the questions have already been answered by Fabio. So Fabio, would you like to read any question?
Yes. Hi everyone. So yeah, I was just typing one. That's why I was a bit slow there. But yeah, so first question was about whether there are some pride data in DBfetch. I've replied already to Ivo. So basically, currently pride database is not supported or not provided in DBfetch, but that's something that we can actually, have a look at and see whether it's possible to add. And as I mentioned there, basically, mostly DBfetch mostly covers sequence data that are used in tools such as NCBI Blast and FastA for sequence similarity search. But yes, we can have a look for sure. So the second question, which I was also typing down, was about the client, the Python client, whether we could get to display a description of each supported database. Oh, okay. Maybe I misunderstood the question. now that I read it again. So what I was replying there was to say that the clients, both the Perl and the Python client, can give you a list of available databases to start with. And for a particular database, they can give you also the available formats as well as the styles for a particular format. So you basically can grow the command to give you basically the formats as well as the styles. A description of the supported databases, I think that's part of the first of the actual method, which is get supported databases. So if you just hit that, it will give you a list of all the databases with a small description in front of it. I think that's what that command returns. So hopefully, if I'm not wrong, I mean, yeah, we can always double check and just run the command. I don't know if it's something that I can actually do now, or that I should do now. But so is that okay? There's a few more questions there. So yeah, probably not a good time to just go and mess with the client. Maybe we can do that together later on. But yeah, so I'm gonna go and read the other questions now. So there was, I suppose, a reply from Ivo saying, "Thank you for your response. I wonder whether in the future DBfetch could retrieve data from multiple databases to be able to do meta-analysis." Okay, that just is gone. Okay, such as okay. Oh, multiomics. Okay, I see. It's in a separate line. So I was trying to find out. Yes, I mean, at the moment, if you use the upload box that Nandana showed in the web page, the databases there need to be, basically all the entries need to be of the same database. But if you use the clients, you can actually mix them. So you basically can put, different databases with the respective identifiers that you are looking for. And in that case, you can get sort of different entries. What is not supported as yet is actually to be able to get sequences or entries in different formats at the same time. So at the minute, you can mix databases with their, corresponding identifiers that you need, but all on the same format. So for example, if you need all the entries in FASTA format or in, EMBL format, that would work, but not really mixing different formats. So maybe that's something that we can probably try as well later on. That's a good point. But yeah, I mean, it already allows you to do that, and also the batch mode that Nandana mentioned in the client. If you have a very large number of entries, the batch mode will allow you to actually, split the work into several different requests so that it doesn't overload kind of the. There's actually a R limit to 2,000 entries at a time, so you would not be able to download more than 2,000 entries at the minute. So you'd need to use the batch mode to actually download more. But the client handles that for you, which is quite nice. So maybe I can put this one as answered live, which it is also belongs to. The other question I'm seeing here is from Mikhail. Will the Python client be available on PyPy Conda rather than for download in GitHub? Yeah, at the minute is not sort of a module as such, so that's why it's not in PyPy or in Conda. It could be easily be provided in Conda, although the folks from Conda also are a bit picky sometimes to what packages or modules they allow there. So that sort of BioConda would be probably the place to be. So yeah, something that I can just make a note to have a look at. So I mean, at the minute, you can basically download them from GitHub, and yeah, that's sort of the way we provide. But yeah, that's good point. So I wouldn't say for PyPy, but definitely for Conda or BioConda, that could be an option. Thanks for the question, Mikhail. Let me put that one as answered live. So I've got another one from Abdallah. I hope I'm saying it correctly. Hi there, thanks for the question. So where can I get ENA sequence ID? So that's a slightly different question. So yeah, in that case, you need to know sort of the entry that you need. So for example, if you have a gene of interest or coding sequence of interest, you actually need already to know the identifier. So I suppose you can just browse the sequences from ENA website, I suppose in this case. And once you get sort of the sequence that you want, you will be able to find out the identifier that they use, and that should work in DBfetch. Yeah. that should be, yeah, that should sort of be the way. But I mean, DBfetch expects you to know both sort of the database that you want to look the data from, as well as the actual sort of session numbers or identifiers that are used on that particular database. I'm going to put that as answer live as well. Okay, next question. Are DB names case sensitive? Yes, that is true. So the page that Nandana showed, which this documentation page, the databases page, which has got all these different examples with IDs and different formats and styles. In that page, you can see sort of the actual database name that is used. And actually, in fact, there are aliases for some of the databases. For example, in UniProt KB, you could use simply UniProt or even SwissProt or Tremble. So there are aliases that can be used, but we don't have them documented properly in this page that Nandan is showing now. So I would say pretty much use the ones that are shown there. And then it's just showing an example there for ENA coding. So that's the DB name, and it's actually case sensitive. If you try, uppercase, it wouldn't work, I think. So I'm gonna type that as live. Thanks, Karsten, for the question. The bit from Evo about the multi-omics, I think it's from the other one. So how many entries can we retrieve with fetch batch? I don't think there's a hard limit there. Thanks, Israel, from the question. I don't think there's a hard limit there. Just bear in mind, if you have, like, say, 1 million, might just take a little while to actually, because it will do like I'm not sure if it's 200 or so that it does it in one go. So yeah, it will just take a little while to actually get. And then at some point you need to be worried about, if you have, depending on the type of output, if you have actually space in your disk. For example, EMBL format for 1 million entries is actually something that takes quite a lot of disk space. So I mean, yeah, it really depends on what you're doing. But faster sequences, for example, you could download easily 1 million into a single file, and that should be fine. It's not, that should be around maybe 200 megabytes or something. So that should be fine. Yeah, so I don't think there's a hard limit for batch mode. Yeah, I'm gonna put that as answer live. Okay, June replied saying that they are not case sensitive, but I think they are case sensitive. So there's a bit of a conflicting answer there. And then can you try? Can you try with? Yeah, I don't remember now. I think they are case sensitive.
Yeah, I can do that.
Just let's just get this answer correct, because if I'm saying it's case sensitive and it's not, then it's. Can try Campbell, for example, just the first one.
Okay.
Yeah. That example, if you try, is this the database name? So if you tried Campbell with just either all uppercase or a mix, or try that.
Yeah.
Oh, okay. Yeah, my mistake. Yeah, sorry. I really thought they were case sensitive. That's good, actually. Sorry. Thanks, Karsten, for the question, and sorry that I was wrong the previous time. And thanks, Jen, for actually getting it right. Let me put a thumbs up there. Good. Next question from Sijal. I'm not sure if I'm pronouncing it correctly. Is it possible to create links between databases like we do in NCBI EU tools that we think? Let me see if I understand. I don't think I know what you're talking about, so I'm not sure I can actually answer. Links between, so is that sort of, are you looking for cross references? If that's the question, then I can say something, but if I'm wrong, then make it. So Sijal, if you could just type down a bit more on how that works in NCBI EU tools, then yeah, maybe you can answer properly. So another question here is about, can we get expression data as well from different databases, expression data? So for example, expression, atlas and such. And I don't think so, because it's same thing as the proteomics data. It's mostly sequences that we provide at the minute. We don't have expression or that type of data in DBfetch, I'm afraid. So I don't think so. We can have a look for sure. Is that so? The reason DBfetch mostly focused on sequence data I'm already mentioned is that we use we basically these same databases are provided in tools such as NCBI Blast and FastA for sequence similarity search. But I mean, obviously, there are other types of data that we could provide with DBfetch. Some of the outputs are sometimes a bit awkward. They're like sort of compressed files or objects or things like that, which, I mean, sometimes it's not easy to add to DBfetch because of that. But yeah, definitely another type of data. I mean, alongside with Pride, Expression Atlas, and these types of resources is something that we can definitely investigate whether to see whether we can add to DBfetch. Yeah. Is it possible to use DBfetch to extract all the molecule processing features from a particular organism in UniProt? No, not really. I'm afraid not. Yeah. No. Yeah. It's. Yeah, I mentioned before. Yeah, pretty much just sequence data, and you then basically, if for example you want the features, you need to download them in SwissProt format, which has got sort of the annotations, and then you need some sort of processing on those to get the features out. You could definitely run a proteome, using the batch mode with fetch batch, but then you'd get all the SwissProt format entries, and from there you'd need some sort of scripting to get just the features, the feature table out. Yeah. Thanks for the questions again. Thank you. And then what else have we got? So another question here, slightly off topic from Karsten. Is DBfetch supposed to replace the EMBL EBI Sparkle endpoint? It's been unresponsive lately. Yeah, no, it's completely unrelated. It's not supposed to replace it. And I don't really know why Sparkle is unresponsive lately. I would say just contact support at EBI so we can get to you, Yeah, sorry about that. And thanks for the question as well. Sorry. And Israel just wrote here, 2 million, thanks. Okay, thanks Israel. Not a question there, so yeah. Answer live. Let me see if I got something. Yeah, I mean, I only can really reply unless I'm missing some question here. Can you guys double check? I think I missed one from Sijal, just because I really don't know how the NCBI utils the links between databases. I'm not really sure how does that look.
I think Sejal has put her question right now. It's at the bottom.
Sorry?
With Sejal's question. So it is basically cross-referencing, for example.
Okay. I was missing that one, yeah. Sorry, thanks. Yeah, it's basically cross-referencing, retrieving all ref-seq proteins and coding sequences for a tax ID. Yeah, I mean, it doesn't support that type of mechanism, but I see what you mean, yeah. So I would say what we have more or less similar to that is EBI search, which provides endpoints. They provide an API with endpoints for cross-references, and they support pretty much all the domains that we provide here in DBfetch, plus additional ones, including like all the Pride and Expression Atlas and things like those that we mentioned before. Because the EBI search really covers pretty much all of the EBI resources. So yeah, have a look at EBI search, and they provide endpoints for cross-referencing, which would allow you to get that type of information. Yeah. We actually use EBI search cross-references ourselves as well for the tools to enrich the outputs. Yeah. Thanks for the question, Sijal. Yeah, I was missing the second part now. Thank you. Yeah.
That's great. Thank you, Fabio. Thank you for.
No worries. Yeah.
And thanks, Nanda, again for your presentation. And yeah, so that's the end of today's webinar. So thank you again, all.